about yourself, like that you can decide is that. Then you go ahead with that.
Good afternoon everyone, my name is Shubham Singh. I am a PhD scholar from Department of Biochemistry and Biotechnology. So here I am presenting my work at the Institutions Innovation Council on development of a detection protocol for circulating tumor cells and drug profile analysis using multi-drug resistance cancer model. So my work basically focuses on liquid biopsy. So we know that regular biopsy of any cancer or tumor type requires too much invasion. We get the sample, it is very painful and risky, and it takes too long to get the analysis results. But with the liquid biopsy, we can go for minimally invasive techniques, real-time monitoring, and also get the predictive analysis for any tumor. So circulating tumor cells are the main focus point of our research. They detach from primary tumors and circulate in blood to look for other nodes and other places where they establish themselves as the secondary tumor, and these are one of the main reasons for metastasis. So here we focus on the detection method. Using many detection methods have been used, have been reported for these circulating tumor cells, such as immunomagnetic separation, microfluidic devices, material cellulation molecule-based enrichment, and nanotechnology-related systems. With normal, both with magnetic properties. So here, as you can see in our analysis, the spectroscopic analysis presents the normal magnetic nanoparticles and nanoparticles with silicon oxide enrichment. These are our results for characterization, zeta potential, size distribution. Here we can see that size distribution, zeta potential, cell analysis show very good size of our nanoparticles. Additional characterization show that physicochemical characteristics of like elemental analysis, crystal patterns. We can see that different both have iron peaks in here with the other nanoparticles showing silicon peak as well, which shows that silicon oxide enrichment was successful. So additionally, we did the biocompatibility analysis. So you can see here that with MCF7, SKBR and BMP3 cells, the cell viability assay showed very good results with our nanoparticles at varying concentration from 1.625 microgram per ml up to 100 microgram per ml. And same for the other nanoparticles as well. Both showed very good results, especially in the SKBR3 cells. This is cell spiking assay, which essentially shows that how many cells were enriched with our nanoparticles. So we can see here that for MCF7 and SKBR cells at 0.2 milligram per ml concentration, the cell spiking showed the capture efficiency was the highest. And in the captured cell, you can see that most of the cells were captured which are present in our system were captured with, as we move forward, less cells, like per ml, less cells were there, which were uncaptured. Finally, here we show our immunofluorescence based analysis. We mainly targeted FCAM molecules there. So you can see that tumor cells with the green fluorescence, which is FCAM immunofluorescence, DAPI, blue color, which presents that it is the viable cell attached to nuclear, and the merged image. And for detection of CTC, we additionally went for CD45 antibody. So our CTC lag in the CD45 expression. So here in the healthy mice, we can see that CTC do not show any immunofluorescence, which shows that it is absent. But CD45 is showing, which means CTC negative and CD45 positive denotes the healthy mice cells. The tumor bearing xenograft mice on the other hand, showed that CTC expression was there, green, which shows the academic immunofluorescence, but it was CD45 negative, which confirmed our results that the CTCs were in fact detected through the immunofluorescence. So these are the results of our primary analysis. We liquid biopsy has many advantages, and with the CTCs having low frequencies, it is a very potent marker to detect any further metastatic potential of our cancers. We hope to translate our research to a diagnostic kit development, and we are progressing with our research. Thank you.
Myself is SAKTHIVEL from Department of Plant Pathology, Faculty of Agriculture. I am here to present my idea on the demo day of presentations of IIC, organized by Institutions Innovation Council. Hereby, I am going to present my seminar on the type of biosurfactant for the effective plant disease management and sustainable agriculture. Why we are choosing this topic means in previous year, that is in the year 2023, severe crop losses or crop losses will be caused by some of the viral, fungal and bacterial plant pathogens. Such pathogens are conventionally controlled using the synthetic chemicals, which pose severe health hazards and some other related problems to humans and to the environment. And that's why we are choosing the early blight pathogen, which is in the tomato, posing severe yield loss to the crop and to the farming community. And these are some of the characteristics of the fungi Alternaria, which can cause early blight of tomato. This is the fruit symptom, which is initiated in the tomato fruit due to the fungi Alternaria. The fruit symptoms including rotting, which spreads, and the bull eye ring spot is the typical characteristic symptoms, and ultimately it leads to the dark circles and finally leads to the shrinkage of the fruits. And in the leaves, the symptom starts with the spotty nature and it grows into concentric ring, which is the typical characteristic symptom for the identification of the disease early blight in the tomato crop, and then the color changes and it appears like blighted and burnt, and a little sharp hole is formed in the center of the spot. This is the artificial culture of the fungi Alternaria solani, which can cause the early blight of tomato. It is typically with the velvety touch and the shades of grey is the typical characteristic identific features for the culture of Alternaria solani. And this is the spore. Spore is nothing but the seed of the fungi, which is generally club-shaped, pale brown in color, multicellular, usually smooth, and it ranges up to 20 to 50 micrometer in length. And in our work, we just control this disease using the biosurfactant rhamnolipids. The rhamnolipids are nothing but the secondary metabolic compound which is extracted from the microorganism, especially from Pseudomonas aeruginosa. Pseudomonas aeruginosa is nothing but a beneficial bacteria which resides in the soil, and we just isolate the Pseudomonas aeruginosa bacteria using several techniques, including serial dilution technique. These are the symptoms of the pathogen, including fruit symptom, leaf symptom, and the auxiliary culture means nothing but the pure culture of the pathogen, fungi, and the conidia. Conidia is nothing but the spore of the fungi. And in the opposite side, we have the rhamnolipids, that is the secondary metabolic compound isolated from the bacteria Pseudomonas aeruginosa. We initially screened nearly 10 Pseudomonas aeruginosa strains, and we selected the virulent Pseudomonas aeruginosa isolate from this, and from this virulent Pseudomonas aeruginosa isolate, we extracted the secondary metabolic component named granulolipids. Using the granulolipids, the in vitro and the in vivo studies are conducted. The in vitro studies is nothing but the studies which were conducted in our lab, and the in vivo studies are nothing but the field experiments. The field experiments includes both the pot culture and the field culture studies. The granulolipids were applied in the various concentration, including 50 microgram per liter, 75 microgram per liter, and 100 microgram per liter, using the poison food technique adopted by Narendra Kumar et al. In 2017. In the in vitro condition, it is clearly shown that the 75 mg per liter of granulolipids application effectively controls the Alternaria fungi growth in the in vitro, and similarly from this observation, we just take the 75 mg per liter of granulolipids application both in the pot culture studies and in the field culture studies. And the pot culture was conducted in the Department of Plant Pathology, and the field culture studies was conducted in the Kadalur district, Pardur village. And now, these are some of the challenges faced by the farmers while managing the Alternaria blight. Continuous application of the synthetic fungicides leads to residual effects, and it also poses severe health hazards to the humans, and it also leads to the degradation of the crop produce and the soil, and which may lead to the fungi to develop a fungicidal resistance. So ultimately for the farmers it is difficult for them to control the disease as it leads to them the economic burden. Now we are just harnessing the power of the granulolipids, the secondary metabolites, against this Alternaria. Granulolipids inhibit the fungal growth and thus serves as an ecologically friendly and sustainable sound solution for the disease management. They offer an alternative to the chemical fungicides, minimizes the ecological impact, and besides the plant disease management, granulolipids also fosters and improve the quality of the plant growth and the plant produce also. And in the first three images we just placed is nothing but the innate immune response upon the application of the granulolipids in the tomato crops shows that the treatment T2, that is the best treatment, 75 mg per liter, shows the enhances the increase in the upsurgence of the innate defense response against the plant pathogen. And in the second row, these are some of the mycelial and the spore image of the Alternaria fungi in the culture plate. Spore is nothing but the seed of fungi, and mycelia is nothing but the branch of fungi. The poison food technique using the granulolipid shows the distorted growth of the mycelium and the spore of the fungi, which is clearly evident from the three photos which are given below. And these are the treatment schedules that were employed during our studies. The treatment T1, T2, T3, we applied granulipids at various concentration of 50, 75 and 100 microgram per liter. And we just compare this, the effect of the granulipids against the commercial fungicides azoxystrobin. Why we are choosing azoxystrobin fungicide means, which is the popular fungicide which is available right now in the Indian market for the control of various diseases, and the treatment for serves as a control. In the benefit cost of ratio point of view also, the treatment to granulipids at 75 mg per liter serves as best, with the benefit cost ratio of 2.67 rupees for 1 rupee. And finally, I am here to conclude that the treatment 75 mg per liter of granulipids controls the disease incidence to the maximum than all other treatments, and it is ecologically sound, sustainable and safe for human consumptions and also for the plants. Thank you.
Thank you. First slide boy. First model. இல்லை வெறும் ஃபோட்டோ எடுத்துக்கலாம். இல்லை இல்லை 3 மணிக்கே போகிறேன்.
Good afternoon to all present here. So today we are going to present the demo practice for the animation presented and organized by Institutions Innovation Council. So our title is the ASHA Freedom to Hope, a promising application to promote mental health literacy and resilience among adolescents to prevent suicides. So usually in India, around 300 deaths occurred per day where adolescents commit suicide. So we wanted to know how mental health literacy and resilience are related in preventing suicides. Already a lot of research has been carried out in the education field where they have given the highest correlation for the mental health literacy and resilience to prevent suicide. So what is mental health literacy? Mental health literacy is nothing but the knowledge about mental health disorders and illness and the attitude towards mental health seeking support, and also the ability to have the efficacy of seeking the help from mental health professionals is mental health literacy. And resilience refers to positive adaptation or the ability to maintain or regain mental health despite experiencing adversity. So how these both are correlated? When a person is having mental health literacy, it is obvious that the person will be resilient to the challenges he is going to face. So to prove this, we have conducted a research in the urban and rural areas where we have selected 200 samples of adolescents aged from 10 to 19 years, irrespective of their education background, and we have conducted a survey method where we have analyzed mental health literacy and resilience of them, and using Pearson correlation we found that they are having the highest correlation, and further giving us the strength to build this app where we can provide these two domains. So the rationale of the study is first, our application should be traditional and modern. In traditional way, it should address the same properties as well as in modern ways. And first, it should address mental health literacy and it should promote resilience and it using the harnessing technology, and in way it should prevent suicides. So these are the objectives of this study. Then the methodology we have established for the first study of our groundwork is descriptive studies, and we wanted to see the efficacy of our app. For that, we are going to conduct 2 experimental studies where we will be analyzing the effectiveness of the ASHA app after pre and post test interventions. So in our findings, we found that there is no significant difference between genders, but there is a significant difference between the geographical locations. And this is the QR code where you can scan and analyze the prototype of the app and how it can work. You can see. Thank you.
We are from the Department of Electronics and Communication, Faculty of Engineering and Technology. For past 15 years, more than 45,000 people have died under the debris in a building collapsed area. The rescue operation of a collapsed building area can be executed in a day to several days, and this will increase the mortality rates. And our idea is to detect the human alive under the debris by using the CO2 concentration levels, thermal images, voice recognition techniques, and infrared radiation which was emitted by the human beings. So our aim is to decrease the mortality rates. Now, already the existing systems are, NASA developed a product named Finder. It is used to detect the people in another planet using microwave reflection. In 2015, earthquake occurred in Nepal. Finder reached the location after 5 days and rescued only 2 people. So since Finder is not a product, it is a prototype with high expensive signal processing.
Technologies used in our device are gas sensor. It is experimentally verified that CO2 gas sensor concentration under the confined environment will be 1200 to 1500 ppm with alive, but the normal temperature concentration will be 600 ppm. Thermal imaging. It images the people under the debris in different colors. Sound sensor is used to recognize the voice by the human beings under the debris. We use passive infrared sensor which detects the human body. If human body emits IR rays that can be detected by PAR sensor which detects the living human body. GSM and GPS. This device used to transmit the information about the human under the debris. This is our system model. The inputs of our microcontroller are gas sensor, thermal image, PAR state, and a sound, a voice from the human body. If any one of these sensors detects the presence of human being under the debris, GSM sends the message to the rescue team so that they can be easily rescued. We have estimated the cost of our device would be around 40,000. We are trying to implement with the robotics, without human. Our device is handled by the human to the place. To avoid this, we are trying to implement and working on it to get a better device. This is our model. This is a working model we have designed. These are some references we have referred to do this project. Thank you.
Hey, the AU animation you can save as animation. No, the slide I skip on it. Yeah, but skip on it. I solely skip on it. You went up. Yeah, fine. What did you have? Say.
But I have
Save as one. Oh.
AU animation matter.
That's what it is.
Sorry, Kannada sir. Sorry. Start பண்ணலாமா? Okay, can start. Okay. Good evening, one and all. My research topic is the immigrating eco-empathy by adopting the synergy of bioplug technology in Indian terrain. India has over 718 districts. Out of the 718 districts, third of the 2 we are facing the day by day we are facing the water crisis problem. So in this respect, we need a tremendous technology for reuse and treatment the water, and every Indian citizen has a responsibility to safeguard the water and to avoid the pollution to the water. In this respect, we are creating the Jalastri water network water conservation network. The lot of conservation technique is available in India, but all the technique is not in an active mode. Some obstacles is there. The really people are interested to conserve the water, but they have some obstacles. So we need to avoid the obstacles. We are trying to avoid the obstacles. That's why we are collaborate that the bioflood technology we are introducing in our technology. So we need a green periods. We know that the green periods need a biodiversity and climate change mitigation, environmental justice and sustainable development and mental well-being. So in this respect, we are introducing the bioflood technology. Bioflood technology is not a new innovation, but the bioflood technology generally the people are doing in a large scale. We are coming into the compact scale. The bioflood technology is one of the aquaculture system which is recycling the waste nutrient as a fish feed, and it's maintain the quality of the water, and it will be culture the microbial protein in the water. So we can see the three pictures. The first picture is the pearl culture. It's the farmer culture the pearl from the backyard of the house. The second picture reveals the bioflak concept. I already told about the bioflak concept. The third one is when we are going to the large scale, we need to supply the aeration to the fishes or any aquaculture system. We need to supply the aeration. So in this respect, when we need to the large scale, the electricity is need. So that's why we are employing the gasoline direct generator. We can change that if we need to the large scale, we can change the gasoline generator as a solar panels or solar motor. Here the generator generating the power electricity. While we are going to the solar, that will be help to reduce the CO emission from the gasoline generator. So our innovation is the Jalastreak tank. The Jalastreak tank is our innovation. It's a portable one. We can generally the bioflak culture, the people culturing the fish and sheep in everywhere, in a backyard of the house what are the places they have. This is our Jalastreak water conservation network system works. The first picture is the colony and a household. We are collecting the wastewater from the colonies and a household. We have two tanks, that is mentioned as the JS tank, that's our innovation Jalastreak water tank. The JS tanks, one is the sedimentation process and the filtration process. Another one is we sediment the water and treated the water. After treatment, we check the quality of the water. If we can do the culture or not in this water, it's affordable for the culture. So after that, we are collecting the water, the pure water collecting in the bioflak culture. We started to the shrimp or fish, any other cultures. So bioflak technology is the minimal or zero water exchange or minimal water exchange process. We don't know what to change periodically the water, because the one time you fill the water, we can get the 3 times of the crop. So we can produce a lot of water from the treatment process. The remaining water will be supplied to the nearby catchment areas. Our extreme goal is to conserve the water will be achieved in this way. So another two tanks is our Jalastreak tanks. One is both the tanks are placed in the backyard of the house. The third picture one lady is that it's a large scale bioflak technology. So these are all the bioflak technology. Our center portable tank is our innovation, and another two is already it's available in the market. Here we already did one pilot study by using the bioflak technology. We taking the 3 buckets of water, which is the water collected from the waste RO machine. Generally we know that the RO machine gives the 1 liter of water to drink, which emits 2 liters of water waste. So we are collecting the waste water from the RO machine, the household. We started the post larva, and we started the culture in the shrimp. Our total investment is 45,012 rupees. Our minimum return on investment is 1,23,738 rupees. We can get the 3 years per annum. We can get the 3 years per annum. Within a 3 bucket of water, we can earn 3,71,000 per annum. We already did this pilot study collected from the wastewater. So these are all the techniques. In the bioflak technology, for normal aquaculture system, there is a connection between the soil and water. But in the bioflak technology, we have cut the connectivity between the water and the ground. So if maybe the people are not interested to the culture, the shrimp or fish or anything aquaculture, we are suggested to the people they can use the Jalastreak tank as to gardening and vegetable races. Here we can see the 2 pictures. One is the thrown 10 rupees bottle, one ornamental plant is there. Another one picture is a RO plant. We already told the RO plant gives 1 liter of water, it emits the 2 liter wastewater. So we are collecting the 2 liter of water from the waste thrown 10 rupees bottle. We bought the 1 ornamental plant from the shop, which is 50 rupees. We bring into the pot, which is grew into the 6. We sold out in the market, each is 50 rupees. Our return investment is 300 rupees. It's a simple concept. While we are going, while we are talking about simply the conservation, water conservation, the people are having started. They are in the routine life, mechanical life. If we are going to the under structure or any with earning with profit, it will be giving a efficient result. Finally, the Jalasteel tank technology gives the opportunity. It's a novelty of the Jalasteel tank is we can employ the tank in everywhere, in every place, as well as it's a lowering cost. The low cost technology, it's create the opportunity to the unemployed people also. It's try to achieve the blue economy and government UNDP and Pradhan Mantri EYMMS schemes also. While we are using our Jalasteel tank, it will be giving the earning, and we can adapting the people who are really interested on conserving the water. It started from the students, senior citizens, graduates, and at all. Finally, our innovation of Jalasteel tanks. Jala means water. The Sanskrit Sri means the word comes from the Sanskrit, which means by asset. The Jalasteel is water is our asset. We need to conserve it. Thank you.
I am Sudarshan Rameshwar from the Department of Chemical Engineering, Annamalai University. So today I will be presenting on what we did in the Smart India Hackathon conducted by Lovely Professional University and the Ministry of Jal Shakti, Government of India. So about our project, what we did is, we did a low-cost desalination technology which can be used in the island like places such as Lakshadweep and Andaman Nicobar Island. So where we have a lot of resource on saline water, but not in potable and drinking water resources. So we prepared a low-cost desalination technique where we will be using a membrane technology in order to propose a solution for this problem. So the idea and approach of our project is we will be using we will be fabricating a membrane which are derived from a biological sources, mainly waste biological sources and low-cost biological sources such as agricultural waste, farm waste and domestic waste, etc. So we will be taking those kind of waste material and will be separating cellulose, biocellulose material in order to prepare a cellulose acetate membrane, which will be low cost and have properties mimicking halophytes. Halophytes are like organisms which found in saline water condition. So we will be preparing a membrane which will mimic these kinds of halophyte organisms. So our material, that the membrane which we fabricated, have properties such as anti-fouling properties, negative zeta potential, and have higher structural stability, and it is low cost. So we will be seeing about low cost, cost effective methods for fabricating these kinds of materials, and as well as eco-friendly methods which are adhere to the Sustainable Development Goal implemented by the United Nations organization. And we will also be conducting a techno-economic assessment and life cycle analysis for these membranes and materials in order to find whether it is cost effective or not. So here we will be using natural inspired biomembrane and various kinds of biomasses such as algal biomasses, bacterial and lignocellulosic biomasses. So here we will be seeing about various types of biological feedstock which can be used for the preparation of these kinds of membrane. The feedstock can be classified into 3 types. We have first generation feedstock, second generation feedstock and third generation feedstock. Here the first generation feedstock contains cellulosic materials, cellulose-rich material such as we have sugarcane, bagasse, sugar beet, soya bean, and other agricultural waste. In second generation feedstock, we have materials like biomass which are enriched with lignocellulose. So we have woody biomasses such as wood chips and forest bases, etc. These second generation biomass can be used for biofuel production such as biobutanol, bioethanol, etc. And third generation feedstock, which is a new kind of feedstock which we can develop in laboratories at large scale, such as algae and other microorganisms, which can be modified to have high content of cellulose and lignocellulose. So these are like third generation feedstock is like a future scope of ours. So in this project we will be using first generation feedstock, which contains high amount of cellulose. That is, we will be using materials such as corn cobs and coconut shells for the preparation of cellulose. And this is the flow chart of our project. At the right side corner, you can see the seashore. From the seashore, our plant will be located like a minimum location distance is 0.5 to 3.5 kilometer. I will tell you why we kept it at that distance. First, we will be taking the seawater into a drum filter, which has a mesh size around 4 into 4 mm. Then the water where the debris, large debris like shoes and other substances will be removed. From that we will be giving it to a sand filter where we like remove impurities such as dust particle and etc. From that the salt water will goes to the membrane, which has a property like electronegativity property, which repels the salt ion, that is positive salt ion and also negative salt ion, and therefore we will be getting fresh water. And like these are all the membranes which are commercially used. The membrane we prepared is PVDF plus cellulose acetate membrane, which the properties are bioderived, ionized with amine and sulfonate group that mimics the mangrove tree properties of repelling sea waters. The advantages of our membrane is it is low cost, about 2.5 times less cost than the commercial membrane. For instance, the membrane which is used in seawater reverse osmosis by Dow Chemical cost around like 50,000 rupees, but the membrane we prepared cost only 16,000 rupees. And that disadvantages is right now we are still in the preparations scale, but we haven't gone for any large scale operation. So which is we don't know what happens if we go for large scale operation. And other commercial membranes are like polyamide membrane, PVDF, that is polyvinyl fluoride, and cellulose triacetate membrane. Then about our IICA approaches, as we all know, Lakshadweep is an island country, island state, island union territory, let's say that, and which has around 36 islands and home to a population around 64,000 people, and the water scarcity is high in that place because of lack of fresh water. So we need some kind of project or innovation in order to solve these kinds of problem in any be it any island territory or island country. So we have prepared a solution where we will be preparing a low cost membrane that can effectively treat and work as a desalination technology, thereby offering fresh water and potable water for these kinds of territory. So our project aligns with sustainable development goals and the goal which implemented by UN, be it carbon neutrality goals and etc. So the thing is, we will be using bio-based membrane prepared from cellulosic, lignocellulosic and algal biomasses, making it cost effective for industrial application and for people who need these kinds of technology. So about our team, my team, the team Graphene of Annamalai University consists of myself, Sudarshan Rameshwar, Krishna, Ehram, Nadia, Hari Prasad and Hari Guru, and we were guided by our beloved teacher and staff, Dr.C.S. of our department, that is chemical department. So thank you.